Hello everyone, today I would like to introduce a work recently published by our team in the journal iMeta, titled Multiomics Analysis Reveals Selection Signals and Loci Characteristics Associated with Complex Traits in Pigs. This research was completed by 40 researchers from 19 scientific research institutions at home and abroad. In the past 100 years, Western pig breeds have been strongly selected for economic traits such as body weight, growth rate, and intramuscular fat content, showing significant differences compared with Eastern pig breeds. The discovery of key genes associated with phenotypic diversification has accelerated the progress of pig breeding, but the genetic basis of complex traits and phenotypic differentiation in pigs is still unclear. Based on 9 genomes and 1,081 resequenced genomes, we constructed a pan-genome and identified non-redundant non-reference sequences and genetic variants. By integrating 162 skeletal muscle transcriptomes and 162 DNA methylation data from 27 developmental stages, the regulatory mechanisms of phenotypic differentiation between Eastern and Western breeds were explored. Finally, a variety of functional experiments were performed to verify the marker genes. We performed Hi-Fi sequencing and Hi-C sequencing and assembled 7 chromosome-level genomes. By integrating the reference genome and the Liyuan pig genome previously reported by our team, a high-resolution pan-genome was constructed, and a total of 134.24 megabits of non-redundant non-reference sequences were obtained. The reference genome, together with the obtained non-redundant non-reference sequences, was used as the final reference genome for subsequent analysis. Next, we collected whole genome sequencing data from 1,081 individuals, which were mainly distributed in Europe and Asia, to do population structure and potential selection signatures. Among the putative genes under selection, we found several candidate genes reported in previous studies. Go enrichment analysis showed that the traits driven by the EAD sweep genes were related to the fat type and reproduction categories, while the genes selected in the WED group were mainly involved in the growth, anatomy, and conformation categories. We then ultimately retained 990 nearly fixed coding variants for subsequent studies. Notably, we found that the coding mutation G965A in BRCA1 gene had an allele replacement effect of 3.55 kg on the 240-day slaughter weight, and increased Eastern pigs' fat deposition. Furthermore, we found a C to T mutation. The selected region of the ABCA3 gene affects immune response and lung damage in pigs. Comparing 27 stages of muscle development in Yongsheng, fat type, and Landrace, lean type, pigs revealed asynchronous growth. Prenatally, LDR genes drove cell proliferation, while TC genes promoted differentiation. Postnatally, LDR genes supported development, and TC genes linked to immunity. Key myogenesis genes were highly expressed, highlighting biological roles and timing differences. Using whole genome bisulfite sequencing of skeletal muscle across 27 developmental stages, we found that Yongsheng pigs had higher overall methylation levels than Landrace pigs. We also found that sequence environments under natural and artificial selection can reshape the overall expression pattern throughout development due to DNA methylation changes. We found a 272 BP insertion upstream of the BDH1 gene in the Eastern pig breed, which showed that the enhancer activity of the insertion was increased. The expression level of BDH1 in Chinese local pig breeds was higher than that in LDR breeds. Further gene overexpression/inhibition analysis showed that the gene enhanced the proliferation ability of myoblasts, but weakened differentiation. The embryonic GHSR gene showed advanced progression in LDR compared to TC. GHSR promotes myoblast proliferation but inhibits differentiation and fusion. The T allele displayed higher enhancer activity, delaying GHSR expression in TC. This SNP reduced slaughter weight by 4.22 kg, with T allele carriers exhibiting significantly lower weights. This study built a pig pan-genome from 9 genomes and 1081 datasets, revealing epigenetic regulation and artificial selection shaping muscle growth and meat quality. Multiomics analysis identified key genes for fat deposition, muscle growth, and immunity, providing valuable insights and markers for breeding programs.